Okay, thank you. So another talk now from the University of Liverpool by Hannah Goldswain, investigating the phenotype of the P323L substitution through transcriptomics and proteomics. Slides.
Thanks. Hi, can you hear me okay? Yes, I'm going to be talking about the P323L substitution in NSP12 in SARS-CoV-2. So early on in 2020 in the pandemic, there was an early mutation diverging from the reference sequence, which was D614G in the spike protein. This was associated with increased transmissibility of the virus. And P323L is a mutation that occurred concurrently with this and is in the RNA-dependent RNA polymerase. And what we found, as you can see in this graph, is that we found that L323 has been maintained over the 4 years of the pandemic at dominant in the population, with some minor variants, but it's been dominant. So we wanted to look at what the genotype to phenotype consequences of this mutation might be. In other studies, it's been suggested that it might interact with host cell proteins. So we did some transcriptomics to look at the differential gene expression. And what we found, we conducted transcriptomics in Cali-3 human lung cell lines, looked at 17 hours post-infection. We found that the transcriptomic profiles of the P and the L, which you can see in the pink and the yellow, are different to the time zero and MOX, and that they also overlap. As you can see in the volcano plots, there's not that much difference between the P versus L at time 17, but there are some differentially expressed genes. As you can see in the heat map, the different groups cluster. So you can see that the MOC at time 17 in green at the top clusters, along with all the time zeros, and that P and L are different. This shows the top 50 differentially expressed genes, and a lot of these are related to the immune system. We found that there were 7 significant differentially expressed genes that are related to autophagy and phosphorylation pathways. We also looked at the proteomics to see if there was any difference in protein expression, but we didn't find any differences at time point 17. So here we used 3 replicates, and you can see that the proteome profiles do overlap, but in the volcano plots you can see that there's no significant differences, but there are differences to the MOC. If you'd like to see more, I'll be at poster 14. Thank you.